All right. Welcome, everyone, to a new episode of the Roscoe's Wetsuit Podcast. I am your host, Toby Passman. On the show with me today, we have a special guest. We have Dr. Dasa Zeithamova. Dr. Dasa is a cognitive neuroscientist and memory researcher at the University of Oregon. She basically studies how memory is used flexibly to draw upon multiple relevant experiences to anticipate future judgments. The Brain and Memory Lab, where her lab operates out of, studies how memories are formed and how they are linked to each other to create internal representations of the world that can guide our behavior. They investigate how different memory systems are implemented in the brain, how they represent information, and how they interact. In the quest for discovery, they rely upon computer-based experiments, cognitive models of behavior, and advanced functional MRI methods. Her research focuses on how we build complex knowledge representations, such as schemas, cognitive maps, or concepts from simple learning experiences. Stacking memories as building blocks, we form knowledge that transcends direct experience, allowing us to use the memory from the past to guide behavior in the future. Dr. Dasa is especially interested in how the hippocampus, a brain structure critical for memory for individual events in our lives, interacts with the prefrontal cortex and other memory systems to support the flexible use of experience. Her primary research tools include computer-based experiments, formal models of behavior, and advanced functional MRI methods. Dr. Dasa, welcome to the show.
Thank you. I'm happy to be here.
Absolutely. So tell me a little, maybe we could just start off kind of what your lab is sort of focusing on right now. What sort of projects are you guys, at least pre-COVID and once everything gets back to normal, what are you guys going to be back to working on?
So I guess we have a couple of lines of research, but the big picture is what you introduced is in how not we just remember individual experiences from our lives, but then how we can then use it to build more complex knowledge. Because after all, it's kind of fun to remember, your last visit with a friend or your wedding or other kind of significant events in your life, but really we probably do not have memory to remember specific past events, but to be able to accumulate information over time so we can then make better decisions and kind of predict what's going to happen next. So most of the projects we are doing in the lab kind of revolve around that. And so one line of research focuses on how we learn new concepts. So essentially, for example, how do you know that this new furry animal passing by is a dog? Have you is it similar to previous past dog that you've encountered, or did you form some kind of abstract concept what a dog is that you can then use? So that's, for example, a line of research where we can use these formal computational models that can help us guess what people are using when they are learning new concepts that we make up in the lab, since we don't know what dogs you've met in your life, so we have to make our own new concepts. So there we can kind of make a best guess from your behavior, what is it that you are relying on, and then we can also look in the brain and see are your brain signals more consistent with a prediction of one model or another model. So that's one line of research, which was interesting because we found out that sometimes we can find the signatures in the brain of kind of both types of representations. To simplify it, it's not dogs, but that you are both referring to the specific dogs that you've met before, and also you've build this general knowledge what a dog is, and you are kind of tapping into that as well.
Sorry to interrupt you, but can you tell me a little what's happening in the brain How are we actually forming these representations? Like with the example of a dog, what brain structures are involved, and how does that take place?
So, so essentially, a categorization task, which is, you see a whole bunch of examples of a given concept, and then you see new things that you haven't seen before, and you have to decide whether it belongs to that concept or not, have been traditionally studied kind of in isolation from memory for specific past events, because it was thought to be independent of the hippocampus, the structure that allows you to remember specific events. So what we are finding instead, that at least in healthy brain, the hippocampus is still involved, and it seems to be doing kind of both things. It seems to be remember the specific dogs, again, our new artificial dogs that we create in the lab, but it's also building the generalized representation of the concept. And what does that mean? That's something that we are still sort of after, but you can imagine that maybe you see, again, going with the example of a dog, you see a whole bunch of different colors of dogs and a whole bunch of different sizes. But what you kind of extract over the exemplars, over the different dogs that you've seen, is kind of the more critical features. Maybe they don't have all possible colors, they have a limited set of colors, but the color doesn't necessarily differentiate a dog from a cat. Then you learn that maybe size also is not perfectly diagnostic. If it has the size of an elephant, well, that's not a dog, but it has certain sizes. So you build, at least in kind of the current working hypothesis, you essentially build this average perfect dog that doesn't really exist out there in the world, but embodies all the perfect doggy features, the furry and barky and, fetches the stick. It embodies the perfect dogginess. And that's kind of that abstract representation, the ideal that you kind of build by taking all the characteristic features and sort of putting them together, although they've never been necessarily present together in any one particular dog that you've encountered. So that's kind of what we currently see. And hippocampus seems to be doing both. It holds on those specific dogs, but also helps to build this generalized representation. You also ask kind of elsewhere in the brain, of course, hippocampus doesn't do it alone. So we are now finding that we have multiple regions in the cortex. So I'm just gonna scooch over so you can see more my hippocampus behind me. So the little blue structure is kind of deep in the brain, the hippocampus. And then cortex is kind of the wiggly, the wrinkly part on the surface of the brain. So we are also looking at different cortical structures, and we are finding that the anterior hippocampus, anterior portion of the hippocampus, interacts more with parts of the cortex that have been in other research implicated in memory generalization, like schemas, your general idea of how a visit to a restaurant should look like, or semantic memory, your general idea of what is the meaning of words. And posterior hippocampus, that seems to be more holding on those specific memories, is interacting more with cortical regions that have been previously shown to be important for memory specificity, like your ability to tell your Roscoe's Vetsuit podcast 1 from podcast 2, what were the differences between the speakers and so on. Kind of this keeping similar things apart versus putting similar things together. And there seems to be kind of a nice division of labor in the cortex, and then the hippocampus does both, but seem to be doing in distinct kind of portions of it.
As you've been explaining this, it's reminded me a lot of the idea of, or I guess sort of the theory of like the grandmother cell, or I think it's what else they call it, like the Jennifer Aniston neuron, right? Is there a lot of support for that theory? My understanding of it sort of being that there's an individual cell or maybe a network of cells that are sort of they are responsible for the representation of some kind of visual stimuli. Is that sort of what you're talking about? Like say with a dog, where you might have a specific neuron or set of neurons that are in charge of processing that information and store that information?
So so in general, the concept that's, you know, external things are somehow represented in the brain is search for an engram. So engram is the neural impression of that external world or representation of the physical substrate of a memory.
You were just starting to talk about enneagrams.
Yeah, so I think that you were asking me about the grandmother, how it relates to grandmother neurons.
Yeah, exactly.
Yeah. So essentially the grandmother neuron view, it doesn't really matter whether we are talking about specific, like how my brain represents Toby himself, right, specifically, versus how my brain represents dogs in general, or what the book is, or what an actor is, or what Jennifer Aniston specifically is. But the idea is that for every item or person or object or place, or for every general concept, we essentially have one neuron that is optimally tuned for it. And that's kind of, you know, really happy when we see that specific thing, and relatively quiet otherwise. It's shocking how well this works when, you know, people poke in the brain and do single cell recording. But at the same time, nobody kind of assumes that this is truly how an engram works, right? As an engram being the physical substrate of the representation. Because the probability that if you poke around the billion neurons, you happen to find the one Jennifer Aniston neuron is really small. So I think that we look this fMRI, if it was really just driven by one neuron, we would never be able to see it because the resolution is very coarse because it's 2 millimeter by 2 millimeter by 2 millimeter, let's say. But in terms of neurons, it's thousands and thousands of neurons. So if one was driving it, we would never see it. So I think that in reality, we think that that's, you know, although it's a good way to look at neurons, in reality, brain seems to have distributed code. So it's kind of the pattern over this, you know, hundreds or thousands of neurons that probably represents, oh, you know, this is Toby, the host of the show, and or this is Dasha, or this is a dog, and it doesn't really matter where in the brain it is. And this is also how we often analyze some of the data. We are looking at even pattern across voxels. So we are now looking over pattern across, you know, each little unit being now thousands of neurons, and we still look at, you know, it's relatively higher here and relatively lower here. And this is the specific kind of signature of looking at a dog, and this is the specific signature of looking at Jennifer Aniston, and this is the specific signature. And that seems to be working better than looking for that one perfectly tuned neuron or perfectly tuned voxel. Right. This is not really related to my research, but if you are interested in the topic, there is a really cool research from Google, which essentially looked at, you know, they have these deep neural networks that they train on a number of images, and object recognition from visual images is now tremendously improved in the last essentially 10 years, although it was almost unsolvable up to then. Like human immediately recognize a dog, even if it's, you know, half covered with other stuff, while the computers really were struggling. So now that there's been a breakthrough, and so Google looked at what is it that allows you to kind of classify the dog and a tree and the sun in images automatically now. And then they looked at those kind of neural, that a classifier can be thought as neural network. So they looked at kind of the different layers, and they found kind of the well-behaving artificial neurons, for example, that were really tuned to the sun and were active every time that you see sun, and not active when you don't on the image. And they deleted it, right? Because it's a computer program, you can just delete it. Nothing really happens. The network was perfectly happy to classify sun just as well. But then they had a whole bunch of, they kind of were systematically deleting differently behaving neurons. And they found out sometimes they delete this very confusing neuron that doesn't behave very well. They have no idea why is it happy for this set of images and not happy for this other set of images. But now suddenly the network wasn't able to classify sun anymore. So kind of this single cell recording, poking around, looking at, oh, can I find a neuron that best responds to this? And now let's study what its behavior. Maybe we were kind of like barking at the wrong tree with this approach, because maybe that's not the most important neuron in the brain to actually solve the task. So,
right.
Not quite my research, but I totally love this little piece, which really kind of shows like we can find these grandmother neurons, but maybe their function, you know, maybe they are not consequential actually.
Right. Well, tell me a little about, so your main tool of research, or your main kind of instrument is the FMRI, correct? Mm-hmm. So tell me a little how that enables you to study memory in the ways that you're looking at. Like how do you utilize that tool?
Mm-hmm. So kind of couple of the, so obviously kind of the traditionally you would have invite people in an MRI session, and then they are solving a task, doing some kind of a memory, such a memory game while they are in the scanner, and we record their brain. And what we are looking at is correlation between what is happening in their brain and what is kind of happening in the task. What the couple of tools that kind of go a little bit beyond that we are using is, one is model-based MRI and the other is pattern information analysis, and I'll try to make it less jargony. So essentially in the model-based MRI, that's the research that allows us to tell apart whether a person is accessing kind of the memories of specific dogs that they've encountered versus the ideal perfect dog that they kind of extracted. So we use cognitive models because there has been a prototype versus exemplar debate for many decades, essentially since 1968, when prototype theory was proposed by Mike Posner, who actually happens to be professor emeritus at the University of Oregon, who kind of came up with the idea that what people learn when they are learning new concept isn't necessarily the specific example you give them, but they form a central tendency, or they extract the central tendency, and that's what represents the concept. So since then, in essentially the 40, 50 years since then, people came up with very formal mathematical models of what kind of response probabilities should be if person is accessing kind of the specific dogs versus the ideal average or perfect dog, and what should be easy and what should be difficult kind of to classify. And so we are using these models, and not only we can fit it in the behavior, we can also predict what should happen in the brain if a person is accessing one representation, the specific dogs, or the other representation, the prototype, the perfect dog. And this has been really fruitful because in the behavior you can only see one model fits better than the other, but it doesn't mean that people didn't also form kind of the other representation, and that's what we are kind of finding. So we are finding that even though people's behavior indicates that they are relying on that, you know, perfect average dog that they somehow find as a summary representation of all the dogs that we've shown them, they actually do also rely on the specific representations, and we can see it in distinct parts of the brain, signal being kind of consistent with the idea. They're also kind of consulting that representation.
And
Does it make sense?
It does. It does. So how similarly does this kind of function of memory work between individual to individual? Like, is there a lot of variability with what you see from one fMRI to another, even like when it's within the same task, or does it generally show up in the same ways?
Yeah, that's a great question. So unfortunately, fMRI is pretty noisy because imagine that people actually do not care just about our task. They may be thinking about what they are going to cook for dinner, or why did they have the argument with their friend before they came to our task. So there is a lot going on in the brain at all times, even if there is no external task. So our task makes teeny bit of dent on, or a teeny bit of an effect in their brain, but there is a lot of noise. So that's one source where just because a scan is different from person to person, we cannot quite make the conclusion that it's a sensible individual difference, because it can be just the difference of what else was going on in their mind during that time. But we are seeing some seemingly systematic differences in behavior between individuals, and we are seeing some systematic differences in what is easy for some people and difficult for other people. So we actually really are, one of the studies that is currently on hold is a large individual difference study, where we want to really have the power to be able to look for these systematic differences. So some people really are perfectly fit by the prototype model and poorly fit by the exemplar model. And others, a minority of people actually seem to be using the specific examples and not form the generalized representation. So right now it's kind of, we don't have enough power to know whether those are like real differences that, carry out to other parts of their life, at least cognitive life. So we want to kind of tackle that. So have, a few hundred people behaviorally tested in multiple generalization tasks, multiple memory specificity tasks, and see if there's really like signatures, there are subgroups, or there are stable individual differences that we can then track what is happening in the brain. So far we have some hints. For example, people who are not well fit behaviorally by the prototype model don't seem to have that prototype. So the prototype model would be the one which assumes that you formed the ideal dog. They don't have really strong signal in one of the key regions where we are seeing the prototypes, which is kind of it's called ventromedial prefrontal cortex. It's in the, kind of if you would touch the very front of the brain and then maybe kind of above your nose, go a little bit deeper there. And we don't see as much signature of kind of these abstract representations being there. So it's something that we're really interested in, is do people have different strategies? Is maybe some people's concept of a dog very different from other people's concept of a dog? And we know that this is true in extreme cases. For example, in autism, autistic people can have really really good memory for specific things, but they simply don't generalize. So I'm having like a starting collaboration with researchers in the Georgetown University who will be looking kind of at how does this work in autism, and do they simply use very different representation of the world than kind of typical people.
Right. Are there any ways, either strategies, or I'm even like thinking like, are there any do we know of anything basically that would enhance these aspects of memory, that you could sort of, like an average person could train? Do we know how to do that yet, or?
No, I don't think we know how to do that yet. We know that it can be made easier or more difficult. So kind of so in concept learning, for example, if we show them typical examples, it's much easier of a concept, something that is not too far from the prototype. People can extract prototype really well. If we show them something more atypical, it becomes really, really difficult. And it's interesting because if I show you a whole bunch of atypical dogs, a week later you may not be even able to recognize those atypical dogs, as even though you were trained on them. We told you these are dogs. But if we show you the typical ones, you can actually then abstract and generalize to the atypical ones much better. So kind of how the task is structured, that can help people to make those generalizations. Another paradigm which we've been using is, let's say, inference task, memory inference task. So for example, you learn that A relates to B and B relates to C. And now can you make the connection that A relates to C? And we can help it if, for example, you encounter the AB relationship and the BC relationship in similar context, then it's much easier to make the connection. So there are some ways where we can we know some knobs that we can turn to make it more likely or less likely for people to succeed in generalization, but we don't know yet whether there is much we can do to kind of change the individual differences or help make it a more generalizable trait that would kind of work in other contexts.
Have you noticed anything in regards to sleep or sleep deprivation and someone going through one of these tasks? Like would that significantly lower their ability to perform one of these memory tasks?
Yeah, so we don't do sleep studies in my lab, but in general, research has shown that sleep plays a huge role, especially kind of when you sleep on it, you get the gist. That's so it's much easier, for example, let's say the prototype, while the memory for specific examples is deteriorating, the memory or your ability to make the connection and kind of form the general idea that can actually improve with sleep. So you can be better the next day than you were the day you were learning. So this definitely, when I'm teaching intro psych, I always tell it to the students, right? Bring your prior knowledge in. That's gonna make it easier to learn, and make sure you always sleep well and sleep before the test, and don't just cram before the exam all night long, because then you will not be able to remember. Right. So yeah, sleep definitely affects memory, but it also disproportionately affects your ability to link things, related things kind of into more coherent knowledge. And sometimes kind of the consolidation during sleep is the thing that helps you make those connections.
Right. I think I also saw some research a few years ago about even naps, how if a student, say studies and then takes a nap and then takes the test, compared to if they were just to go about their day without a nap, that, there's improved scores. I'm not sure if it was specifically pertaining to memory tasks, although I think it may have been, but I'm not positive on that.
It definitely has one of the strongest effects on memory, because what does going about your life do is you are just encountering more and more information which will interfere with what you just learned. So the NAB helps prevent the interference of, the new information, makes you kind of push aside the old information that you learned before that.
Right. And how does the brain how do we decide upon, obviously we would go crazy if we were able to actually remember like every single face that we saw, if we're in a big city, if we saw, remembered every single face that we ever encountered throughout the whole day, we'd go crazy. Our brains they selectively remember what hopefully needs to be remembered. How does that, how does that occur where our brain is able to like pick what sort of stuff to remember?
What to prioritize? Yeah, I think that you hit a big question, kind of what I guess it's the next 100 years of memory research going to be focused on. But you also mentioned the faces, so I'll try to link it to another example of the methods, the FMRI methods that we are using that are a little bit different than this model-based, and try to kind of lump it in my answer. So, so first, if something is related to what you already know, it's much easier to remember. So you will remember better the faces that in some way perhaps are familiar. Young people will better remember young faces, and will maybe just generally represent the older faces as, oh, that was an older gentleman, and then in a lineup, they may no longer be able to tell which older gentleman this was that they've met. But they may be able to remember which specific new, young college age face it was, if they are college students. When something is emotionally loaded, that will definitely be better remembered. So if perhaps, you felt attracted to the face, or you felt threatened by the facial expression of the person, that all can kind of heighten, improve your memory. So if it's in some way relevant to you, or you will need this person later, it may boost your memory. So those are kind of all the little things that can decide what we will forget and what we will remember. One thing that is not necessarily often talked about is that essentially a lot of this is already happening way earlier than memory. It's happening at attention. You simply sometimes do not pay attention to the faces of the people you pass by. You don't really focus on the face to encode the features. And I don't mean encode as in like intentionally trying to remember, but you never really paid attention to the face in the first place. So maybe even the perception wasn't really there, and therefore the memory cannot be there. So that's happening as well. Not related to your question, but related to your earlier question about other ways how we use MRI. I was mentioning a little bit that instead of kind of looking for that perfectly tuned neuron, we are now looking at kind of patterns of activation, and maybe we have one experiment with faces where we are looking at how do you represent different faces that you encountered, and essentially how do you represent the identity of the face, and then how do you represent, if we teach you certain concept, let's say these three faces belong to one family, how do you start representing that? And kind of the way we do it is, let's say if we have a patch of cortex or patch of brain, we looked at kind of activation across a range of neurons, and we look at how similar the activation to this phase looks to the activation to the next phase that we show them, right? Are they represented similarly? Are they represented differently? And we can see kind of what if a part of the brain is representing certain faces similarly and other faces dissimilarly, what are the features that that part of the brain cares about? What we were surprised to find that learning affects representations almost everywhere in the brain where we look. So if we show participants' faces and tell them, okay, this is Peter Miller, this is Kyle Miller, George Miller, Paul Davis, and so on, and we have 3 last names that we kind of reuse. We can ask people behaviorally how similar these 2 faces look to you, and they will start rating after they learn the family names, the faces from the same family as more similar than physically equally similar faces that are from different families. And we manipulate the faces, the physical similarity between faces, by creating them as a blend from, real faces. So we now can make them more similar than they would have naturally been. And when we looked at what's happening in the brain, we assumed that kind of these maybe generalization regions will show, increased similarity after learning, kind of representing the whole concept of a Miller family. Instead, we are seeing it everywhere. Visual cortex starts representing 2 Millers as more similar than 2 faces that we made, artificially made equally similar, but we give them different last names. So that was kind of an exciting, unexpected, but exciting recent finding, which really was only possible by using the fMRI in this other way, rather than looking at, what's on and what's off. It's kind of what's the specific way how this part of the brain represents the face, what is it highlighting? And looks like the whole brain starts highlighting the more relevant features, the ones that go with kind of the commonalities that are defining the family, than other features that may be in common but are not related to the family concepts.
Sure. Well, awesome. Dr. Dasha, we're coming up onto the end of the show. I really enjoyed our discussion today. If people want to find out more information about your work or your research lab, where would you direct them to?
So my lab website is cognem at uoregon.edu. The cognem is a shortcut for cognitive neuroscience of memory. So COGNEM, cognitive neuroscience of memory, .uoregon for University of Oregon.edu. So there is a link to our papers, which of course are, take a couple of years to get out. So you can also check out our posters that we presented at conferences recently. That's kind of more current snapshot of what we are up to. And they can also email me at dasha@uoregon.edu. The first name is just DASA at uoregon.edu.
Awesome. And for those listeners who enjoyed the show today, go ahead and like and subscribe to our YouTube channel. We're Roscoe's Wetsuit. And you can also find audio versions of the podcast available on Spotify, Apple Podcasts, Stitcher, and just about anywhere else you can listen to audio podcasts. Also, go ahead and check out, we just launched Roscoe's Wetsuit Premium, which you can find at patreon.com/Roscoe's Wetsuit. So go ahead and check that out. There's going to be some exclusive content on there. Dr. Dasha, again, thank you so much for coming on the show today. Really enjoyed our conversation.
Well, thanks for inviting me. It was a pleasure.
Awesome.